Researchers have developed a new fluorescent label that gives a clearer picture of how DNA architecture is disrupted in cancer cells. The findings could improve cancer diagnoses for patients and classification of future cancer risk. Published today in Science Advances, the study found that the DNA-binding dye performed well in processed clinical tissue samples and generated high-quality images via super-resolution fluorescence microscopy. My lab is focused on developing microscopy techniques to visualize the invisible, said senior author Yang Liu, Ph.D., Associate Professor of Medicine and Bioengineering at the University of Pittsburgh. We are one of the first groups to explore the capabilities of super-resolution microscopy in the clinical realm. Previously, we improved its throughput and robustness for analysis of clinical cancer samples. This has bothered me for more than 10 years, said Liu, who is also a member of the UPMC Hillman Cancer Center. A molecule of interest is labeled with a special fluorescent dye that flashes on and off like a blinking star. When several images are overlaid, the complete picture can be reconstructed, at a much higher resolution than previously possible. Until now, the problem was that fluorescent dyes didn't work well on DNA or in processed clinical cancer samples. After showing that the new label produced higher resolution images than other dyes, the researchers compared colorectal tissue from normal, precancerous and cancerous lesions. The images show that as cancer progresses, chromatin becomes less densely packed, and the compact structure at the nuclear border is severely disrupted. While these findings indicate that the new label can distinguish normal tissue from precancerous and cancerous lesions, Liu said that super-resolution microscopy is unlikely to replace traditional microscopes for such routine clinical diagnoses. Instead, this technology could shine in risk stratification. Early-stage lesions can have very different clinical outcomes, said Liu. Some people develop cancer very quickly, and others stay at the precursor stage for a long time. Stratifying cancer risk is a major challenge in cancer prevention. To see if chromatin structure could hold clues about future cancer risk, Liu and her team evaluated patients with Lynch syndrome, a heritable condition that increases the risk of several cancer types, including colon cancer. They looked at non-cancerous colorectal tissue from healthy people without Lynch syndrome and Lynch patients with or without a personal history of cancer. The differences were striking. We need to follow these patients over time to measure outcomes, but we're pretty excited that chromatin disruption in normal cells could potentially predict cancer risk. In future work, Liu and her team are interested in examining chromatin structure in endometrial tissue from Lynch patients, who The researchers also received funding recently to look at sputum samples from smokers for early detection of lung cancer. Other authors who contributed to this study were Jianchuan Xu, Ph.D., Shuijiao Sun, M.S., Hongchang Ma, Ph.D., Rhonda M. Brand, Ph.D., Douglas Hartman, M.D., and Randall E. Brand, M.D., all of Pitt, and Mingfang Bai, Ph.D., and Kwangho Kim, Ph.D., both of Vanderbilt University. Visualizing the invisible. New fluorescent DNA label reveals nanoscopic cancer features. Science Daily. Science Daily, 4 March 2022. ift.tt/tgjnd2p. University of Pittsburgh, 2022, March 4. Visualizing the invisible. New fluorescent DNA label reveals nanoscopic cancer features. Science Daily. Retrieved March 4, 2022 from ift.tt/tgjnd2p University of Pittsburgh. Visualizing the invisible. New fluorescent DNA label reveals nanoscopic cancer features. Science Daily. ift.tt/tgjnd2p, accessed March 4, 2022. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.